In our first video, when we talked about the overview of nucleic acids, we pointed out that DNA has a purpose. Its purpose is to encode the blueprint for proteins. And we go through processes called transcription and translation to make that in effect. In this next video, I'd like to talk about yet another process that occurs with DNA, and that is the process of replication, in which DNA is copied into another copy of itself from double-stranded DNA to double-stranded DNA for propagation of genetic material into progeny organisms, into the next generation. And replication is a very complex process. We're going to focus on the basics and the chemistry of it, and a little less on the biological context of it, as this is a biochemistry class. So I'd like to move into this page where we're going to talk about replication, and we're going to first talk about some of the basics, the overview, and the terminology. Let's just say again that replication is copying DNA to DNA. We're going to make an exact copy of the chromosomes in the organism, whether it's a bacteria or a human cell. Let me tell you that there's some basic principles involved. Replication is semi-conservative. We'll describe that in a minute. It's primer-driven. We'll also address that. And lastly, it's going to be template-dependent. So let's get into the basics. If we're going to replicate DNA, we got to go back to its common structure. Don't forget that DNA is anti-parallel. If we take this cartoon of a single strand of DNA going 5' to 3', remember the other strand must be 5' to 3' in the opposite direction. And the first thing we need to do is form what we call a replication bubble. A replication bubble is when we take the DNA in the middle of some sequence and pull it apart to form two single-stranded regions in an area that looks like a bubble. We could also point out that we end up with two areas where we go from single-stranded to double-stranded and then double-stranded back to single-stranded. We call these replication forks, and these are terms that are used often in other classes and in things you might read. Now, the idea of semi-conservative is that when we tear apart DNA into single strands this way, ultimately we're going to make replication of a new strand with the old strand, and we're going to do so on both sides. And this idea of semi-conservative means that we will end up with two new double-stranded molecules. Here's one of them down here, and here's another up here, and each one of those conserves or maintains one of the original and has one new strand. So that's what semi-conservative means. Primer-driven is a very strange thing, but it's something that's worth describing right now. And that is, when we start the process of copying DNA, we don't do so with making new DNA. We actually use RNA, small pieces of maybe 4 to 5 bases of RNA that are going to be laid down as primers. So we call these RNA primers. And we're going to use these to begin building DNA. Now, what we need to say is that we still have our directionality in an anti-parallel fashion, and it turns out that the new RNA that we just created is going to be anti-parallel as well. What that means is we are going to have this little primer going opposite to the strand that it is annealed or complementary to on the top. And this strand on the bottom is going to be anti-parallel to the DNA strand on the bottom. And what we are going to do now is talk about how we're going to start building DNA. So I'm leaving the RNA red, and I'm going to build the new DNA in blue. And the key is that we are going to build DNA in the 5' to 3' direction. That's what everybody in biology teaches you, and there's a little bit of a problem with that. It makes it a little difficult. When we meet in person and do this, I always stop and have students put arrows, we'll do it in blue here, to show which direction these primers are going to grow if they go in the 5' to 3' direction. Let's have you try that. Think about which way these two primers are going to grow if they obey the rule of growing in the 5' to 3' direction. Okay, I hope you paused and tried that out. And when I do this in class, I usually get at best about 50-50 in terms of the correct answer. And the reason is because when we say you're growing in the 5' to 3' direction, it's not clear if we're talking about the new strand or the strand that we are building on. Now, the DNA strands that we're building on are called templates because we use those to copy. We have two templates here. And then the new strands that are growing are called extending strands. Here's the answer. The answer is that we're going to grow the top strand here to the left, and we're going to grow the bottom strand to the right. That is growing in the 5' 3' direction, and what we mean by that is we are growing from the 5' end of the new strand to the 3' end that continues to extend. Sometimes people think, well, I must be growing in the 5' 3' direction for the template, and they go in the wrong direction. We got to get that correct. And here's a very simple way to make this easy. DNA and RNA, by the way, we'll put that down, always extend off the 3' terminus. What do I mean by that? If we go onto any piece and we find the 5' and the 3' terminus, the 3' is over here, we're always going to extend off that. Same thing down here, 5' here, 3' on the right, we're going to extend off that. If you term it that way, then it forces you to be on the new molecule and forces you to go in the right direction. So that's a critical part, and we are going to leave it there because we've talked about templates, primers, and semi-conservative.